Okay, we start off with the discussion on amino acids, and as we saw last time, the amino acids consider or have rather an asymmetric carbon atom to them. To this carbon atom is attached an amino group and a carboxylic group. Okay, and we have the different side chains. We found out that we have the 20 common amino acids, the 20 side chains, the 20 R side chains, which differ greatly in the property that they have, and we have associated with them a 3-letter code and a 1-letter code. If we go to this amino acid, this side chain corresponds to the amino acid glutamine. The 3-letter code for glutamine is GLN and the 1-letter code is Q. Okay, so, it is something that you ought to remember regarding the amino acids. Now, if you notice in this structure that we have here, that is called a stick structure, in the stick structure we do not see the hydrogens. Okay. The nitrogens are always marked in blue, the oxygen atoms are marked in red, the carbon atoms are marked in green, that is for the asymmetric carbon, and the others are marked in grey. Do you see that clearly? Okay. Now, this is universal. Nitrogen atoms are always in blue, acidic or rather oxygen atoms are always in red and carbon atoms are in grey. Now, the next thing that we looked at was the joining of the two amino acids. The joining of the two amino acids gave rise to what is called the peptide bond. Now, when we look at the peptide bond, we see that we have associated the carboxylic group of the previous amino acid with the amino group of the next amino acid. So, this R group corresponds to the first amino acid of this particular dipeptide, and this R group corresponds to the second amino acid of the dipeptide, which means that in a polypeptide chain you always have the first amino acid begin with an amino terminus. Okay, so, it is never written any other way. You always have the first amino acid in a protein, in a polypeptide chain as the amino terminus. It always is an NH2 terminus, and the carboxylic terminus is always the end of the protein chain. Okay. Now, if we go to the next slide, we just have the one that I showed you earlier, where we have the elimination of water to form the peptide bond, and in this case we have a tripeptide, and from the R groups we can identify the peptide. Okay. Now, you understand that as we have a protein chain or a polypeptide chain, this is going to get longer and longer. Now, as it gets longer, we have here, as you can see, this is glycine because it has 2 hydrogens, this is alanine because it has just the methyl group attached to it, and this is cysteine because it has the SH group attached to it. And we found out also that this cysteine amino acid can form what is called a disulfide linkage. Okay. So, what we saw in the previous slide was the formation of a peptide bond. How is the peptide bond formed again? It is formed by the elimination of water between 2 amino acids. Okay. So, we have the carboxylic group of one amino acid lose its OH with the H of the amino group of the next amino acid, and that is a strong covalent bond that is formed. Okay. The only other covalent bond that is formed in proteins is this disulfide linkage that is formed by the linking of 2 cysteine residues that may be far away in the protein chain to form what is called the disulfide linkage, that is the SS linkage, this SS linkage that we show here. Now, I mentioned about this experiment where the whole protein chain was unfolded and it was found that exactly the same disulfide linkages were formed. Okay. I will just go through that experiment once. Now, when we look at a protein structure, which we will be doing in the other classes to come, this is what is called a native structure, an active native structure. What does an active structure mean? It means that this protein or this enzyme has its function intact. Okay? So, when we are looking at this active enzyme, we know that its enzymatic activity is to the fullest because it is a folded protein. It is in its native structure. Now, if you add beta mercaptoethanol to this, beta mercaptoethanol will reduce the SS linkages to form SH. So, your cysteine SS disulfide bond is now a cysteine residue over again. Okay. So, now we have added beta mercaptoethanol, the structure of which is given below, just the BME that is written here. Then we add urea. What urea does is, we learn again later, is it unfolds the protein. Now, what do we mean by unfolding the protein? It means that all the interactions that occur in the folded protein, which we will be studying as we go along or understanding protein structures, the whole thing is unfolded to form a long polypeptide chain. So, that is what it is going to look like now. Why? Because we had the native structure, we had 4 SS linkages. Now, what happened to those 4 SS linkages? We reduced the SS linkages to form SH. Then what we did is, after we formed the SH, we added urea. Once you add urea, what happens is it unfolds the protein completely. So, now we have this long chain with 8 SH bonds to it. So, what is this? It is a denatured, inactive, random coil. What does a random coil mean? It means it is not folded anymore. It is just like any necklace, so to speak, with beads on it. It is not folded. So, now what happens is, the SHs now are free to link with any other SH. You have 8 SHs. So, number 1 could link with number 5, or it could link with number 6 or with 7. So, now when we look at this structure, we believe that the SHs could practically or possibly link with any others, because there are many such conformations possible. But now what actually happens is, we remove the beta mercaptoethanol. In the removal of BME, what we are ensuring is that these SH linkages, or rather the SS linkages, can now reform. Okay? Because we are now, instead of reducing it, we are oxidizing it. So, if we look at these SH linkages that can form, we understand that since there are many conformations possible, it might form any one. Then what we do is we remove urea. So, what am I doing? I am allowing the formation of SS linkages. I am allowing the protein to refold. Okay? In doing so, it is found that it forms the exact same structure that was there before. What is this structure? It is exactly just one conformation. Okay? And what is this there? It is the native structure of the enzyme that is fully active with the 4 disulfide bonds correct to what was there in the original protein. Okay? Now, if you just think of the whole thing, it is extremely fascinating, considering that this protein has 124 amino acids linked together, right. It has 124 amino acid links linked together, and it had 8 SH linkages, which could have bonded with any other one. I allowed the protein to unfold. So, when I had this unfolded structure, it could have essentially formed any or folded into any conformation, but that does not happen. It always forms this one conformation, that is the native structure of the fully active protein with the exact 4 disulfide bonds correctly linked with one another. Okay? And that is what is extremely intriguing about protein folding, and it is still not understood to date. Okay? Nobody understands how a protein folds into the same structure each and every time. Okay? Now, these are some modified amino acids which you just need to know the names of, considering that these amino acids are present in proteins to some extent. Their names are hydroxyproline. Okay? What is different here is you see there is a specific OH attached to this. Can you see the OH attached to the hydroxyproline? That is one other amino acid not among the common amino acids that you observe in proteins. Another one, the second one, is gamma carboxyglutamate. What is glutamate? Glutamate is when you have the single carboxylic acid group here. If you have another carboxy attached to it, it becomes gamma carboxyglutamate, and this is the structure which you see in some proteins. Another one is phosphoserine linked to the O. What is serine? Serine is, now you recognize this as part of the polypeptide chain. How is this part of the polypeptide chain? Let us just get here and we will show you how this is part of the polypeptide chain. This you see, if we look at this region here, what do we see here? This is the NH of which amino acid? The serine, the phosphoserine in this case. And what is this the part of? It is the carboxylic part of this phosphoserine moiety. So, what is happening next? Here, if I link these two together, the covalent bond or the peptide bond would be this linked to the NH of the next amino acid. Fine. So, this would be your what group? What group is this? Your R group. Okay. So, now you can see that again, if we look at the carboxyglutamate, what do I have here? I have part of the polypeptide chain, and you always notice what is the one that you have on the left? The nitrogen, the amino part of the carboxylic acid always, because that is the way you read the protein. Okay? Because now what you're not always going to get structures, you're going to get letters one after the other. So, what do you have to do? You have to read the sequence of the protein. When you read the sequence of the protein, then you have to recognize the symbols, and these are redundant. What is the information that you need? You only need to know what R is there. That is all you need to know, because if I tell you that I have glycine, alanine, valine, if you know the structures, you can draw the polypeptide chain, right? Because you know how they are linked to one another. You know that the NH2 has to be on the glycine side. The carboxylic part has to be on the valine side, and the alanine would be intermediate like one of these, right? Is that clear? So, that is the way we consider all the amino acids. So, all the information you actually need is a series of letters, the alphabet, the amino acid alphabet that comprises proteins or polypeptide chains. So, what are the modified ones that we could have? There are others, but these are the common ones. We can have hydroxyproline, in which our hydroxy is attached here. We can have carboxyglutamate, where the carboxy is attached here. We can have phosphoserine where the phospho is attached here, the phosphate group to the, instead of the OH. We can have phosphotyrosine. Tyrosine, remember, is one of the aromatic amino acids that we studied the last class. What are the other two? They are phenylalanine and tryptophan. Okay? And so, what do we have here? Instead of the OH that we would have for tyrosine, we have the phosphate attached to it. So, it is now phosphotyrosine. Now, usually when the polypeptide chain is formed, this is not a common occurrence. Okay? All these happen, or all the modified amino acids that you see occur after the protein is formed. Okay? So, these are usually, as I have mentioned, written here, these are modifications that actually occur after the protein is synthesized, and what we call them are post-translational modifications, because the process of translation is how you get from RNA to protein. Okay? What is the central dogma of biology? It is DNA to RNA to protein. The process of DNA going to RNA is known as transcription. The process of RNA going to protein is known as translation. So, after the translation, it means you have RNA translating to the protein. So, you have the synthesized protein. Now, if you have what is called a post-translational modification, you would have these specific modifications that I have shown on this slide that occur in the already synthesized protein. Okay? Now, what we see on this slide are groupings of amino acids. We mentioned some groupings last time, the last time when I showed you the individual structures of the amino acids, but these are extremely important in the properties of the proteins, in the catalysis of the enzymes, in the catalytic properties of the proteins, in their functional properties, in their surface properties of the proteins. The grouping of these amino acids is very important. Now, if you go to different sites or even different books, there are subtle differences in the groupings. But that does not really matter, because what we will be looking at is we will be looking at the gross groupings, meaning that we will be looking at aspartic acid and glutamic acid. These are the acidic amino acids, right? We look at arginine, lysine and histidine. Now, this is where there is one change at times, where the histidine at times occurs in the positive set and at times occurs in the uncharged polar set. Okay? We will see why that occurs in a few slides from now. So, the ones that are definitely negative are aspartic acid and glutamic acid. The ones that are definitely positive are arginine and lysine. Okay? We also see histidine in this group, in this particular grouping of amino acids. Now, what does this mean? If we have a negative amino acid or we have a positive amino acid, these amino acids, as I mentioned earlier, are likely to be on the surface of the protein. Why? Because they are going to interact with the solvent. You know that when you are speaking about the solvent, the protein is where? It is in a solution of physiological pH, that is, say you have the protein in your body, it is in the blood. The blood is primarily a lot of water in there. So, what kind of a solution is it? It is a polar solvent. Okay? So, all of the ones that we are looking in here would favorably interact with the polar solvent. If we look at the second set that we have here, these are uncharged polar. So, where do these charges come from? They come from the specific side chains that they have, and if you have a knowledge of the side chains, you know exactly what they are. Then we have the arginine and the lysine. In the uncharged polar set, you see how we have the changes in these are the 3-letter codes and these are the 1-letter codes. Then we have the non-polar amino acids. These non-polar amino acids are also called hydrophobic amino acids, would tend to be away from the solvent. Why? Because they have no favorable atoms or no favorable interactions with the solvent. Why is that? Because all these side chains mostly contain carbon and hydrogen. Okay? So, what do we have here? We have alanine, we have glycine, we have valine, leucine, isoleucine, proline, phenylalanine, methionine, tryptophan and cysteine. Now, the last 3 of these have heteroatoms in them. Okay? Methionine has a sulfur, tryptophan has a nitrogen and cysteine also has a sulfur. Okay? But since they have a predominant amount of carbon and hydrogen to them, they are put in the non-polar group. Okay? So, where would these favorably be? Unless they have specific reasons to be on the surface, they would rather be buried in the protein. Okay. Now, what we mean by buried in the protein is we will study later when we look at protein structures. Okay. Now, we have to consider specific properties of amino acids. What are these properties? One thing that we have looked at is we have looked at the size and the shape. Shape mean, what do I mean by shape or size? Size means molecular mass. The smallest one is glycine. It just has that hydrogen atom in there. What is the next smallest one? Alanine. Why? Because it has the methyl group. Then gradually we can go, then what is the largest, the most bulkiest one that we could have? Tryptophan is the bulkiest one and the longest chain that is arginine or lysine. Arginine is the longest chain, then we could have lysine. Okay? So, these are the long chain amino acids that you could have. So, the size and the shape of the amino acid is dependent on what? On the R group, because the rest of it is the same. What is the rest of it? We have the asymmetric carbon atom that is referred to as the alpha carbon atom. Then we have the amino group, the carboxylic group and a hydrogen that is common to all amino acids. Then it is only the size and the shape of the R group that differs. What else? The charge. We just saw in the previous slide how we have the groupings. So, we can have positive amino acids. What are the positive amino acids? Lysine and arginine. What are the negative amino acids? Aspartic acid and glutamic acid. Okay? The next would be the polar amino acids. The ones, now what is the difference between the charged ones and the polar ones? The polar ones, preferably some of them being small like serine, can occur in the central part of the proteins. They will form hydrogen bonds because they have an OH to them, like serine has an OH. So, it can preferably form a hydrogen bond. Threonine has an OH that can also preferably form a hydrogen bond. What about the amide ones? What do we have? We have 2 amides. What are they? Asparagine and glutamine. Okay? So, we could have sets of hydrogen bonds formed with them as well. So, we could have them being polar, forming hydrogen bonds, but uncharged. Okay? That is what the difference is. Then we have probably the most important property of amino acids, the hydrophobicity. Okay? That is extremely important because it determines the protein folding nature. Okay? That is what is, we will study a lot of this hydrophobicity as we go along. Another one is the aromaticity. We find 3 amino acids that are aromatic in nature: phenylalanine, tryptophan and tyrosine. Okay? And as I mentioned before in the last class, that it is these 3 aromatic amino acids that contribute to the absorbance of proteins. The optical density or the UV absorbance that you see of proteins is solely due to the aromatic amino acid residues. and this we monitor at 280 nanometers. Okay? That the UV absorbance of proteins is usually monitored at 280 nanometers, and the absorbance or the optical density that is observed is solely due to the presence at 280 nanometers of the aromatic amino acids phenylalanine, tyrosine and tryptophan. Then we have the conformation, that is usually determined by the side chain. What do we mean by the conformation of the amino acid? The side chain, now when I have a long side chain, it has single bonds in it. It is free to rotate in 3-dimensional space. So, I am going to have a conformation associated with the property of the amino acid. If I have an alanine, I have just a methyl group. Okay? So, that will not as such have any conformation because all of it will look identical. But if we have a long side chain like lysine or arginine, we have a lot of single bonds in there. What can happen to those single bonds? They can rotate. As they rotate, say I have the lysine rotate, then what is going to happen? The NH2 group that forms the end of the lysine group will what? That will also rotate, that will also change, and as it changes, it is going to change the conformation of its side chain. This we will study, the propensity to adopt a particular conformation. This comes when we will be doing the secondary structure of proteins, which we will be doing in detail later on, and the relative position in proteins we can say from the properties that we already know. What are these? One is the hydrophobicity. From the hydrophobicity, I can straight away say that, say for example, a phenylalanine or a leucine or an isoleucine is expected to be in the central part of the protein. A lysine, an arginine or an acidic amino acid like aspartic acid or glutamic acid is expected to be where? On the surface of the protein. Okay. Now, this is very important. Histidine. Remember I mentioned 2 slides ago why histidine is sometimes considered to be a positively charged amino acid and sometimes it is not. Okay? The reason is that all of you know what pKa means, right? We will be studying this in a great detail because this is extremely important for protein structure. Now, the side chain that you have here is the side chain for the histidine. Okay? Now, if you notice what histidine can do is it can accept a proton, and as it accepts the proton, the side chain gets protonated, right. So, what has happened here? Apart from my, what is this? This is my asymmetric alpha carbon atom. The nomenclature of this is such that if this is the alpha carbon atom, since the carboxylic acid part and the amino part is common to all amino acids, this carboxylic carbon is always referred to as just C. This C alpha means that the side chain is connected to this carbon atom. Then we have this CH2. What comes after alpha? Beta. So, this is the C beta. Okay? There is a specific nomenclature which we will study as we do protein structure later on, but for now it will suffice if I say that to C alpha is attached C beta of the side chain. Okay? So, alanine will just have a C beta. The next one in this case would be, what would this be? This would be a C gamma. Then we would have 2 deltas, a C delta and an N delta, and so on and so forth. Okay? So, just by the nomenclature alone, you can actually identify which amino acid it belongs to. Why? because the R chains are different for each case. Okay. So, an alanine is just going to have a single C beta. What about valine? It is going to have the C alpha. Every amino acid has a C alpha. What about valine? It is going to have a C beta and 2 C gammas, right? A C gamma 1 and a C gamma 2. Fine. Okay. So, let us get back to our understanding of the protein donor and protein acceptor capabilities of the histidine side chain. The pKa of 6 means that when there is an equilibrium at a certain pH, right, where it can either accept the proton or it can donate the proton. Okay. Now, apart from what is here, okay, which we will be studying in a moment, the ionization of amino acids, at this particular point, I can protonate this nitrogen to form NH plus. It can also deprotonate to form this back. So, this is an equilibrium. Okay. Now, this equilibrium occurs at a pH close or rather between 6 and 7. Okay. Now, if you look at what the physiological pH is, that is the pH of blood, the pH of which all the enzymatic reactions in our body are taking place, that is at a pH of 7.4. Okay. The physiological pH is 7.4. Now, what happens to histidine is that because of this protonation and deprotonation that can occur, histidine forms a large number of the catalytic sites of proteins, okay, just because it can accept and donate a proton close to the physiological pH, which is not the case with the other amino acids. If you are too basic, your pKa is going to be too high. The pH of the body does not reach that amount of protonation or deprotonation. Okay? Because the physiological pH of our blood or where all the reactions are taking place in our body is close to the pKa value of the histidine side chain, this forms a component in many enzymatic reactions, in many catalytic sites of proteins. So, what are we talking about? We are talking about basically the ionization of amino acids. So, what do I mean by an ionization of an amino acid? Initially, if I am at a very low pH, what is going to happen to the carboxylic group? It is going to remain as COOH. What is going to happen to the amine group? It is going to remain as NH3 plus because I have a large amount of protons in solution. Okay. That is exactly what we are going to see here. So, at a pH of 1, when the pH is very low, I have COOH. I also have NH3 plus. We all understand that. Why? Because I have an acidic solution which has sufficient amount of protons to protonate all the side chains or all the amino groups. We are not talking about the side chain right now, but since the amino acid itself has a carboxylic group and an amine group, we can consider the protonation of those two sites alone. So, what do we have here? We have at a pH of 1, we have our carboxylic acid protonated, we have our amine group protonated. There is no other place where a proton can be accepted here. As you increase the pH, okay, or rather if you consider a titration, say you are adding base. So, what is going to happen? The base is going to abstract the protons from the amino acid in the order or in the ease of abstraction. Okay. So, what is the first one that is going to get lost? It is the carboxylic acid one. So, at a pH of 7, I have lost this. So, I am no longer C double bond OOH, I am now COO minus. But the amine group does not lose its proton at this pH, right. I keep on increasing the pH, or rather I keep on adding more base. At a point, I will lose the proton that is attached to the amine group. Okay. So, if I do an ordinary titration of the amino acid, I will get how many inflection points? 2. Why? One corresponding to the loss of the carboxylic acid H, the other corresponding to the amine H plus. So, this is where I get one set. What is this pKa? This is pKa 2. What does that mean? It means that as I close on and reach the pH close to 2, I am going to lose this proton. Okay. This pKa is 10. So, after I reach a pH of 10, will I lose this proton? So, at a pH of 12, I am now at this position, where I have lost all the protons possible in the amino acid. Is that clear? So, initially what did I have? I had everything protonated at a low pH. As I go on, what do I have? I have a form. What is the charge on this form? It is a zwitterionic form. It has a positive charge to it and a negative charge to it. So, it is effectively charge of 0. Okay? That is the zwitterionic form of the amino acid. Every amino acid is going to have this form. Okay? Now, what becomes fascinating is, suppose my R group also has a charge to it. So, where is this going to become interesting? When I have an acidic group or an amine group attached to it. So, this will come into the picture when I have an aspartic acid, a glutamic acid, a lysine, an arginine and the histidine for the pKa 6. If I consider the ionization of the amino acid, I have an equilibrium dissociation constant associated with it. This is true for all acids. What is the equation that we have here? We have HA dissociating into H plus and A minus, and what is the Ka associated with it? It is this equilibrium here, where I have H plus, A minus and HA, right. Now, you all studied about buffers, where we are looking at what is called the Henderson-Hasselbalch equation. What is that equation? You basically rearrange this equation, we rearrange the expression rather, and we write it in such a manner that we get the H plus on the left hand side, right. And I know that the negative logarithm of the hydrogen ion concentration corresponds to the pH. Okay. So, what do I have here? I have the pH of the solution equal to the pKa, right. What is the pKa? It corresponds to this equilibrium, plus log A minus by HA. So, what is this? This is the base form, this is the acid form, or rather the salt form or the acid form. Okay? That is the way we write what is called the Henderson-Hasselbalch equation. And if the concentration of these two are equal, it means I have equal amount of undissociated and dissociated forms, then this is going to have a ratio of 1, making the logarithm 0, and my pH is going to be equal to pKa, right. So, what are we considering here? We are considering our amino acid as 1HA form, right, and we consider the dissociation from HA to H plus and A minus. And what am I getting from there? I am getting a pH equal to pKa when my A minus is equal to the HA, fine. Okay. Now, this is what happens when we titrate an amino acid. Okay. The titration of an amino acid is going to show a diagram like this, which you will understand very, very clearly. First of all, we consider these as ampholytes. Why? Because they have both an acidic and a basic group attached to it. That is true for all amino acids. You have an acidic group, which is the carboxylic acid group. You have a basic group, which is the amino group. So, these are called ampholytes. Now, as the base is added, so this what is here along the x axis, we have number of OH minus equivalents added. So, basically what are you doing? You are adding NaOH to your amino acid solution. Initially, if we make the pH very low, what does my amino acid look like? All of it is protonated. So, I have COH and NH3 plus. Is that clear? Now, what am I doing? I am abstracting H plus. How? By adding OH minus. So, if I add OH minus, I abstract protons. So, there is a dissociation immediately. Okay? As soon as I hit upon this pH, what happens? What do I have at this pH from the previous slide? I have equal amount of this species, which is my A minus, and this species. Now, I want you to understand this very clearly, because you have to remember that as soon as you start OH minus addition, this H plus starts getting abstracted. Clear? So, as soon as I start, the first drop of OH minus does not bring me to the pKa, but it starts abstracting H plus. So, I come to a point, as I keep adding my OH minus, I come to a point where this is equal to this, right? What is that point? That is when my pH is equal to my pKa. That on this graph is this point here. So, the pK1 value, or as it is called, is 2.34. What does this mean? It means that at this point I have equal amount of the carboxylic acid anion here and the COH minus, and the COH I mean, the CO minus and the COH. Now, what I am doing, it is now in its zwitterionic form. I keep on adding OH minus. As I add OH minus further now, what is going to happen? At some point, I am going to lose the amine proton. The NH3 plus has to lose its proton when I cross its pK2, then after I am at this level, say. So, I am at pK1 where I have equal amount. As I keep on adding OH minus now, I have more amount of A minus than HA, right. Now, I keep on going with the titration. Then when I come to this point, what do I have? I have equal amounts of this form, which is now my A minus, and my HA, which is this, right. So, this is my HA because now this is my acid, which has this additional H plus added to it. As I add OH minus, I abstract this proton at a particular pH. The pK2 will correspond to a pH where I have equal amounts of this species and this species. Is that clear? So, now if I keep on adding OH minus, I will just be increasing the pH because I now have just OH minus in solution and this. Is that clear? So, this would be the titration of glycine. You would have two inflection points, the first corresponding to the carboxylic acid proton, the second to the amine proton, right. But now, the fun begins when we do not have H2 here. Suppose I have something else, okay. Suppose I have glutamate. What does glutamate have now? It has an additional COOH to it. So, at what do I have at low pH now? At low pH, look at this species. This is the side chain. So, this is our side chain, right. This is the carboxylic acid part of the amino acid. This is my NH3 plus. At this point, all of them are protonated. Why? Because my pH is very low. So, at my low pH, that is at this region here, all of these are protonated. I am now adding OH minus. What is going to happen? I will start abstracting the proton, or rather the group that will most easily lose its proton will start the equilibration. So, that will be this proton. The proton that belongs to the carboxylic acid of the amino acid, not of the side chain, will be lost first. Okay? So, what is the species that is being formed? It is this. This is still protonated. This is still protonated. Fine. Now, I am continuing with the titration. I still have how many more protons that I could lose? 2. One belonging to the COOH of the side chain and one the NH3 plus proton, right. So, as I go on further with my OH minus addition, my titration, the next easiest one to be removed is the side chain one, right. Now, I keep on going. Now, what is going to happen is I now have to lose the last proton that is left, that is here, the amine group. So, the 3 pK values that I now have correspond to the, what does it correspond to? We have 3 protons that can be lost. So, we have now, that's wrong. Okay. So, what do we have? We have 3 protons that could be lost. We have the first one lost, which is the carboxylic acid group attached to the C alpha of the amino acid, right, not the the side chain. Because why do we lose that first? Because that is the most acidic proton. Then we go on to the next one, which is the next most acidic proton, the side chain one. So, we have the side chain one that we are now going to lose. Then we lose the amine one. So, what do I have in my curve here? I have a pK1 value. I have a pKr value. What is the pKr? R is the side chain, so it corresponds to the side chain. Then we have a pK2 value corresponding to the amine. Okay. Let us do histidine now. When we saw the histidine pKr, remember we showed a pK of 6 where we had the changes or where it could accept a proton or donate a proton. So, that is exactly what is happening here. What do we have? We have now at low pH, what do we have? All the possible sites protonated. So, we have the side chain protonated, we have the amine group of the amino acid protonated, we have the COOH protonated. As we increase the OH minus additions, then what is going to happen is I am going to lose this proton, right. So, as I lose this proton, what is the next one that I am going to lose? The one belonging to the side chain. The last one that I am going to lose is going to be the one belonging to the most basic group, which is the amine group. Okay. So, again I have my profile or my titration curve rather look like this. Is that clear? Okay. So, I have a pK1 corresponding to the COH, I have my pKr corresponding to the side chain, and I have my pK2 corresponding to the amine group, right. Okay. So, what do we have here? For the glycine, I have 2 pK values, right. What is this form? It is the zwitterionic form. So, at a particular point, if I go back, let us just go back 1 minute, this previous slide. Okay, what do we have here is I have a pKr value, but if I go, okay, what do I have here? Here I have something called a Pi value, okay. That is the isoelectric point, okay. Now, what do we mean by an isoelectric point? An isoelectric point is the point at which you would have the zwitterionic form of this amino acid and the charge would be 0. Is the charge 0 at this point? It is. So, what is my Pi value? It is the sum of the pKa of the carboxylic group, the pKa of the amine group by 2. What happens if I have a glutamate? When is the charge 0 for the glutamic acid? The charge is 0 for the glutamic acid at what point? When it is between these two. Now, at the last, see this is pK3 or rather pK2 for the amine, this is pKr, this is pKa. So, it first loses this one, right. So, in between these two my charge is what? 0. So, what is my Pi value? 2.1 plus 4.07 divided by 2.
Net charge of the solution will always 0.
Net charge, it depends on what pH of your solution it is. If you have a pH corresponding to the Pi, then the charge on the amino acid is 0. When we have lysine, what does lysine have? Lysine has an additional NH3 plus, okay, when you do a titration curve, or if you study a titration curve of lysine, it is going to behave differently. And what do we have? We have the pi now at a point where lysine is uncharged, that is between the pKa of the amino group and the pKa of the side chain this time. So, what is the pi of lysine? It is very, very high. Okay. So, to summarize, what we have is we have the ionizations of the amino acids. These are the regions, the low pH regions correspond to the aspartic acid, glutamic acid, the intermediate region to histidine, then we have lysine, and finally we have arginine. Okay. So, these lysine and arginine are the two basic amino acids. Aspartic acid and glutamic acid are two acidic amino acids, and this, as I said, could act as a proton acceptor and proton donor depending on what pH you were at. Okay. So, we will stop here for today, and so the discussion that we had today was on understanding the properties of the amino acids, their size, their charge, their polarity, and the titration of the amino acids, which is extremely important when we consider the ionization that could occur based on how or what the pH of the solution is. Thank you. Okay, we continue our discussion on the acid base chemistry of amino acids, which we were discussing in our last class, and we understood that if we have a structure such as this, as in glycine, we have basically we have our protonated NH3 plus and we have our CO2 minus. Now, this is zero charge, right, because it has reached the point where it has lost the carboxylic H plus, the proton belonging to the COOH, and it is yet to lose its proton that is attached to the amino group of the amino acid. Now, when calculating the pi, you have to remember that you have to go from a +1 to a -1 charge through the 0. Okay. So, for each structure that you are considering, we have to remember that if I want to go from the +1 to the 0 to the -1, these are the two steps that I am going to follow. So, if I want to calculate the pi of glycine, what I have to do is I have to sum up the pK1 and the pK2 or the pKa's of both the acids